So good morning, everyone. Let's start. We were waiting a little bit for people to wake up, actually. And so today we will start with a small session of short oral communications. Then we will continue actually with a coffee break. Then we will have one symposium on evolution of musicality. And that's the program before lunch. So that's it. I think that we can start. Who is the first one from this session? Who wants to start?
Good morning. My name is Beatriz, Codo. I'm from the Laboratory of Neurodevelopment and Evolution from Universidade Federal de Minas Gerais. And I'm going to present a little bit of the work we do in the lab. The title of our work is Dopamine Behind Brain Development and Behavior, an Evolutionary Approach, Perspective, sorry. So we know dopamine is a modulatory neurotransmitter in all vertebrates and is involved in the sensory perception, the regulation of prolactin, regulation of body temperature, is involved in the balance between hunger in society and in sexual behavior. So it's really conserved around all vertebrates in these functions. Dopamine has has 5 receptors in mammals, and when they are activated, there is a molecular pathway that leads to dopamine-dependent behaviors. I'm not going to explore a lot of these here because we are focusing on the behaviors. So we know that dopamine is involved mostly in motivation and aversion behavior, in the reward system, motor behavior, and attention. And that this regulation of dopamine can lead to a lot of neuropsychiatric disorders, especially during the beginning of the development of the animal. So dopamine is present in all metazones, and the evolvability of the dopamine system was really important for our species to keep alive in the evolution. In dopaminergic receptors, they are really similar in sequence, in structure, and function. But the different kinds are not related between each other. Today, we use mostly animal models to study the role of dopamine systems and receptors, especially mice and zebrafish. They are really conserved compared with humans. So we can use these animals to modulate how this happens in the human and make suppositions about how human behavior works. So during development, dopamine has a role in several phases of the brain development. Neuromigration, myelinization, synaptogenesis, apoptosis, and this really, when we have different, when we have disruptions in different phases, we have different behavior consequences. The objective of the work I'm going to present is to evaluate the role of dopamine on behavior in different development windows across distinct animal models. I'm going to present work we did on early development and late development. In late development, we explored the perinatal and postnatal phase. So I start with early development. We use zebrafish. That is a small fish that is used a lot in research of behavior, neuroscience, development, and genetics. We expose the zebrafish larvae to dopamine during the 3rd to the 5 days post-fertilizations. We put the larvae in those little plates. We chose these days because it is important to the development of dopaminergic and GABAergic systems. And we had a group of control larvae that weren't exposed to any drugs. Then we did behavior tests in 5 days post-fertilization, 7 and 14. So the first test we did is the open field test. So it's a test when you put the animal in a in a arena, and you record how the behavior of the animal was. So we measure how much mobile episodes the larva started, how much time the larva spent swimming, and how much the distance travel. It's here. Oh, sorry. So we saw that the chronic exposure to dopamine decreased motor behavior in zebrafish larvae in all ages. We saw that during the in the 5 days post-fertilization was really a strong phenotype of this behavior, but in 14 is a little weaker, so it's recovering a little bit. And we also did the light dark box test. There is a test where there is a space that is light and there is a space that is dark. And the larvae prefers to be in the light side because it feels protected. So it explores less the dark side because it's like it's something aversive. We also call it like anxiety-like behavior because it's phenotype similar to anxiety, but we can call it anxiety because we don't know the emotional state of the animal. So our larvae with 5 days, they spend a lot less time in the dark zones. Spend a lot of time really stopped in the freezing behavior, but this behavior didn't maintain during the rest of development. To explore the late development, I'm going to present first the perinatal phase. That is like the beginning, really beginning after the female gives birth. So we use mice for this behavior, and we injected L-dopa, the precursor of dopamine, in mice, in the little pups, from the 1st day to the 5th day of birth. And we also use a group that received just the vehicle, que was saline, as our control group. We divided by sex, so we make our work with females and males. And we did the behaviors when they were adolescents. So we did the open field, that is a similar idea, then the zebrafish. We did the elevated plus maze. That is attached where you have a maze that is a little elevated from the floor, and 2 arms are closed, have closed the walls, so the animal is protected, and 2 arms have open walls, so the animal can see that is elevated and he can fall. So the principle of the test is when the animal has increased anxiety-like behavior, it avoids the open arms. We also did the feeding suppressed by novelty test. That is a test where we let the animals 24 hours without food, And in the next day, when you put the animal in a new environment, the food is in the middle. So the animal wants to stay protected in the corners, but is also hungry, so he wants to explore the food. And then he has to assess like a balance of explore and taking risks for food. In the open field, we saw that the chronic exposure to dopamine increased the exploratory behavior of male adolescent mice, but we didn't saw anything in the females. Now, just to be clear, the hearing is the exploratory, the vertical exploratory, when the animal is exploring like this. Sorry. In the Elevate Plus maze, we saw that the chronic exposure to dopamine did not change the adverse behavior in adolescent mice in any or females or males. And in the feeding suppressed by novelty, we saw the decreased exposure increased adverse behavior in adolescent mice, males only two. To evaluate the postnatal behavior, we injected dopamine from the second day to the 14th day of after birth, and we did the open field in like that box. Like the box in mice is similar to the zebrafish, only that the mice prefer the dark instead of the light. Okay. So in the open field, we saw the L-dopa females show increased exploratory behavior and decreased adverse behavior when compared with saline. When the animal is in the center, is exploring more, and it also is being less sensitive to adverse behavior, because the animal prefers to stay in the corners, just like humans when we enter a new space. But in the like dark box, we saw that the saline L-dopa the L-dopa females also showed decreased adverse behavior, because they prefer to stay in the light a lot more than females injected with saline. So I did a little like a summary, because it's a lot of data. So we saw that in early development, all larvae had the chronic exposure to dopamine, decreased exploratory behavior, increased adverse behavior. And in the late development, perinatal, we saw increase of exploratory behavior and adverse behavior in males, and increased exploratory behavior and decrease of adverse behavior in females. Okay, but what does it mean? So we saw that dopamine has a role in neurodevelopment that is conserved throughout evolution, but has different roles, because in distinct development windows, we saw different behavioral alterations. Our next steps now is to evaluate the mechanisms, how this is happening in the cell, and also focus on different receptors. So we are now working with Kimperoli and SKF that are agonists of the 1 and the 2 receptors. Eita. Oh. Okay. I just was like to thank for all the organization of the meeting, for allowing me to present our work, and for everybody in the lab that helped to do all this work, and everybody, all the people who gave us money too. So, thank you. Ah, tá, desculpa.
Just one question. I recently saw a TED Talk by Helen Fisher, if you know her on this, and she associated lust, sexual desire, with serotonin, and passion or addiction, whatever, with dopamine, and trust and comfort with oxytocin. And she didn't distinguish between early and late development. I don't know what you think of Helen Fisher's arguments there, but if you have any idea, I'm curious to know what what you think about that.
Just so I'm sure I understand. You want to know like what I think about this sexual behavior? You mean, or the? Sorry.
Eu vou falar em português, acho que é mais fácil, né?
Tá.
O Helen Fisher, né, você se conhece, essa pesquisadora sobre sexualidade, paixão, coisa. Ela, eu recentemente vi uma fala dela no TED Talks, em que ela associou luxúria, desejo sexual, com serotonina. Associou paixão, que ela associa também a obsessão e a ficar viciado em coisas assim, né? Ela falou que isso estava relacionado com dopamina. E depois confiança, conforto, estava associado à oxitocina. Ela não fez diferença entre desenvolvimento...
Entendi.
Queria saber o que tu acha dessas falas dela.
Tipo, eu posso responder em português? Ou... Eu não sei. Não, então eu vou responder em inglês mesmo. Meu inglês não é muito bom, então já peço desculpas. Mas, so dopamine is involved in the addiction and the compulsive behavior. We saw that a lot of drugs they are involved in the activation of dopamine systems, and then the use of the drug changes the balance of receptors. So when the person starts to need even more drug, and then when the person stops to use the drug, the system is all disbalanced because all receptors are altered. So, and we also saw that normally when the mom's addicted, a lot of babies already are born with what's the word? Abstinence. So we know that anything can affect the development of the brain. And I never read specifically about the balance of oxytocin and serotonin, but I think it makes sense to think about dopamine in this, like you mentioned. I don't know if I answered your question. Okay, thank you.
I will do one question only.
Okay.
Do you have a hypothesis on why female mice was less aggressive with dopa? Because I think it's common sense that dopamine is correlated with reward system. So exploratory behavior are very related to dopamine. But I was very interested with the relation between dopamine and aggressiveness.
We didn't measure aggression specifically, but we also know that dopamine is involved in the motor behavior. We saw that when dopaminergic neurons are dying, we have Parkinson. So my hypothesis is that during this increase of dopamine is make this increase in exploratory behavior. But when I see all work in our lab beyond this one, we are noticing that the female development and the male development they are not exactly synchronized. So we saw some effects in males during a window, and in females like a little bit later or a bit little earlier. So we want to see now the behavior in adult mice, because this is adolescent mice, so to see how it would be.
Sorry, I have read aggressive instead of aversive. Sorry.
It's
my
because
mistake.
the test is about anxiety-like behavior, but I feel a little uncomfortable to jump to a decrease of anxiety, because I don't want to assume that my control mice are anxious. So I think about how the mice has a decreased aversive behavior to the environment of the test. So, thank you. Anyone want?
Hello, Andreza. I was thinking about if you know something, I don't really know about the relation of dopamine with personality traits, because the tests you used are all used for measuring personality in animals. So I think you have that data already for exploring the relationship between dopamine and personality and the development of personality. Have you done something like that?
Yeah, I read a lot of work that use this elevate plus maze and they like their box to separate the animals in bold animals and animals that are a little more shy. But I never explored it a lot. But I think it's really interesting. Yeah.
You can use your data already for
yeah, that's true.
Inducing or inferring some relation with personality. I don't know.
Yeah, that's really interesting.
And you already have that data. You can publish.
Thank you. I'll tell the people in the lab. Okay, thank you.
Hello. Good morning. Hi, my name is Andreza. I'm a doctorate student at Federal University of Espírito Santo, located here in Brazil. And Dr. Tokumaru is my advisor, and Josan was my advisor during my doctorate sandwich at University of Bucharest in Romania. So I'm excited to share some of my results from my doctorate, and today I'll focus on the psychometric properties of the evolutionary fitness scale, Brazilian version. So, depression is one of the most common mental health issues around the world, and it's a major cause for disability. The prevalence of depression in Brazilians is 5.8%, and women, individuals aged in 40 and 59 or more than 80 years old, live in urban areas, low education, smoke, and chronic disease are more likely to have depression in Brazil. I'm fast because of the time. So, fitness has been described in evolutionary biology as reproduced successfully and it transmits gene to the next generation, and the situations and behavior that reduce fitness result in psychological pain. So, our hypothesis considers that those who suffer from depression have a fitness problem to be solved. Based on that, the evolutionary fitness scales was developed by Josan in 2018 as a measure for dimensions implicated fitness in humans. Humans suffer from depression. Oh my God. Okay. The scales is composed for 45 in the case the respondent do not have children, or 58 items in the case of the respondent has children. It's scoring the likely type scale from 1 strongly disagree to 5 strongly agree, and the scale was validated in America, thus the original language is English. So our aim was to translate and culturally adapt the evolutionary fitness scale to the Brazilian population and evaluate its psychometric properties. So this was the processing. This study was approved by the Research Ethics Committee of Federal University of Espírito Santo, and then there we followed the process of translate. First, we need to translate and for Portuguese Brazilian speakers, and then back translation to English, and this verse was sent to the original author to evaluate it and make some comments if necessary. Then you test in small sample 12 people who was instructed to do some comments and see if the language was okay, how could be more clear. And then you did the data collections in a large sample, 710 people, use a Lime survey, and this was the measuring: socio-demographic, evolutionary fitness scale, patient health questionnaire 9, the World Health Organization quality of life, and the social adjustment scales. So we did the exploratory fact analysis and then the confirmatory fact analysis. Our sample was the university community, and the criteria was aged 18 years old or over, and then the sample consisted of 715 participants. However, 5 of them were classified as missing data for filling up wrong both control questions, resulting in a final sample of 710 adults. This was the control questions. As you can see, there is just possible one correct answer. I am Napoleon's brother or sister? No. And I have crossed the Atlantic Ocean by swim? No. And the 5 people answer yes. That's funny. I don't know why. Okay, so this is our sample. Most were white, 51.9%, followed by brown, black, Asian, indigenous. Here in Brazil, we have these black people, consider brown, black, and I am, for example, brown, and they have black people. So the mean age was 31.8 years old, most were females, most were single, undergraduate, and 26 had children. Use this factor software for conduct the EFA, that's in order to verify the cultural difference, the structure of EFS. And this factors is a free software produced by Lorenzo Silva and Fernando. So this is the step of exploratory factor analysis. First, we try to do FS, EFA, but in the whole sample, but the software indicated any error in our output. So the best decision to resolve that was divide the sample into 2 groups, those who have children and the total sample. The total sample was used to conduct EFA from the items 1 to 45, and the items 1 to 46 to 58 questions about offspring were used to conduct EFA for those who had children. And items either fully loaded or cross-loaded was on multiple factor were removed. This resulted in a full fact solution with 34 items. Male veil, this is the name of the factors. Male veil, 7 items. Health, 7 items. Social capital, 11 items. And offspring quality, 9 items. The adequacy of the model was evaluated using the KMO test, the root mean square error of approximation, and the comparative fit index, and the Turkel-Luz index. Our results were great. This is the parameter. And then it's our result. As you can see, it's higher than minimal. Then H, it's amazing. It's an index that evaluates how well a set of items represent a common factor, and the higher H values suggest a well-defined latent variable, which is more likely to be stable across the study, which means that EFSBR dimensions, may value have social capital offspring quality, may be stable across studies. And for the reality of the construction, we did the Chromebox alpha that was great as well. Finally, we did the confirmatory effect analysis for test this structural finding in FEA with JASP. This is a free software for University of Amsterdam. And the user recommended cutoffs that indicate a good fitting for validation. This is the parameter, and this is our results. As you can see, it was great as well. So, but, although the Bartley test was, was significant, the result is expected to not be significant. But the other indices supported the model. This p-value was explained by the residual covariance between some pair of items. By inserting this residual covariation to the model, there was an improvement of all fit indices. Bartley test is a very sensitive test, so there is no problem in the model if this is happening. So the local relation between factors supported the multifactorial structure. This is our model, the CFA. As you can see here, we have the items that's for every dimensions, and the decompositive reality determine the variation in the factors load of the items. So we did this article omega for every dimension, and that was great as well. And finally, because the original verse showed a two-factor solution, person adaptedness, factor 1, and factor 2, partners of spring fitness, we test both solutions in our sample. Our solution, finding our sample, the multifactor, and the B dimensions found in the original study. But we did not find any support for two-factor solution. On the other hand, the CFA supported our four-factor solution. About the constructor validated, we were expected to find the third moderate positive correlation between EFS dimensions and the whole quality of life. And this is the result. Every dimension was significant with quality of life. And we expect to negative correlation between EFS dimensions and PHQ-9 for depression. And every dimension was significant except the FES offspring quality. This is the items for mate value. I read just 3. That's the most important thing. I am satisfied with my sex life with my partner and I are very comfortable sexuality. I believe my partner is faithful to me. In the health, I eat very healthy. I am active outdoor person. I exercise at least 4 times a week. My family members, social capital. My family members brag about me. I am admired by my friends, and I am important to people rather than my family. Offspring quality. I have a close relationship with my children. My children's confident in me, and my children trust and follow my advice. High correlation were observed between the 3 EFS dimensions, mate value, health, and social capital, and PHQ-9 for depression. Suggests that the high score in these areas implicated in light depression, and vice versa, supporting our hypothesis. However, there was no correlation between EFS-BR offspring quality and PHQ-9, as expected. We hypothesized that another variable should be taken into consideration. In general, those who have children can be engaged in marriage or a long-term relationship. In our sample, that was true. The major was marriage, and being married can be protective against depression. So, we have here the common precipitants for depression, and here in the side, in the left, in the right, we have the FES-BR dimensions, and we hypothesize that people who suffer from unemployment, maybe it will be a problem with in the social capital, and the lack of social support in postpartum depression. Hi, Hegel. It will be a problem with social capital, and the social rank by price, that's hypothesis for price. It will be a problem with social capital, and the romantic strife with Mayvale, divorce Mayvale, and the grief social capital, and the offspring quality in case of the mother or the father lost her child. And the chronic disease in health. But we need further research to test this hypothesis. And this happened to me as well. So there was a photo here from my lab and my team, my colleagues. And I would like to thank you for the conference and for my colleagues from my lab, and a special thank you for my advisor, Dr. Suemi Tokumaru, and for CAPS, who supported my scholarship, and for the others who helped me. Thank you so much.
Thank you very much. Did you have any items that reflected conflict in social relationships in the fitness scale?
No. No, we have this item that was conflict with health. This was about attractiveness, and there was a crossload together with the health. So to resolve this problem, we cut about, we need to put the high number for cut the items, but you don't know why it was together. But yes, we know, because if you look for the literature and in Western society, health is related with attractive. And healthy people is more able to find them for mate. But no social relationship, I think.
Okay. And did you, just out of an exploratory fashion, look at the association of your fitness scale with the individual symptoms of depression? So you were using the PHQ-9, I think. I'm curious, for example, if you found any interesting associations with suicidality or sadness, or if there are any differences at the symptom level.
Yes, we have a clinical sample, but we have just 50 minutes, so I didn't present the clinical sample, but we have this result. It's very amazing, interesting. And another opportunity you presented this, the clinical sample.
Great, thanks.
Hello, thank you very much for your presentation. I was just curious about the two questions that allowed to take off 5% from the questionnaires about the Napoleon sisters or brother and the other. What is the importance of this question?
Look, when people answer the kind of questionnaire, it's an online questionnaire. They're receiving the homes and they opening the questionnaires and start to answer. So we have a lot of questionnaires there. It was 176 questions. It's a lot. So use the control question to test if the person is really pay attention in the answer, in her answer. So if they answer like this question like that, yes, I'm Napoleon brothers or sister, and I crossed the Atlantic Ocean by swim. This proves for us maybe they is not paying attention correct in the question. So you need to cut off as missing data because you don't know what's happening. So that's maybe that's wrong. This was just match and match. Do not pay attention to the questions, correct?
I understand. These questions were in the original questionnaire, you chose it? Because I don't know, if I were to answer a question like this with many questions, and if I saw a question so obvious, maybe I would be tempted to answer it wrongly, because it's so obvious the answer, you know. So I wondered if maybe it was a possible behavior.
See, it's because that we have first test in a small sample. And in the small sample, we have this control question as well. And we asked for the small sample, how was to answer that? This kind of a question makes sense for you. And received some feedback for this small sample. There was, are you trying induce me to answer that question? No. But the best solution for testing if people is pay attention is put the control questions. And maybe because the age, these 5 people was very young. Maybe they did this because they wanted to do. It's like, oh, this is a funny question, I will put yes. But I don't know, so how can I know? So I don't consider it as a missed data. Thank you. Thank you, Professor Higue as well. Thank you.
Thank you very much for nice presentations. And we will continue with Lilian. Right? Am I right? Okay. Okay.
Hello, everyone. Good morning. Okay, now they told me that I need to stand here, and this is a little difficult for me, but I'm going to try. So. I'm Lilian. I'm a postdoc fellow at Painel Hóspede Gêmeos, the Twin Panels panel. I'm a white female girl with a short hair and a blonde hair. And I'm using black and jeans for those that can see me. So now I'm talking about a little bit of what we are doing using thermography to detect our facial indicators of dental anxiety in twins. Okay. So let's start. Remember that some individuals are more susceptible than others to environmental influences. That's influences that for better or for worse. Some positive influences can be social support or attention, and negatives should be such as injured or overwhelmed state. And these individuals respond to different stimuli. I'm a little bit tired. So let's think about the possibility of going to the dentistry. So now you have to go to the dentist. How do you feel about this? Do you feel anxious or afraid? So let's remember the differences again of anxiety. That's an emotional state preceding something that you're expecting, and afraid And fear is a response to an object or a situation that you are facing. So we are working with anxiety. It's here. So, okay. So fight or flight, okay. If we need facing a stressful situation, we present some behavioral and physiological changes. And these changes are something like the, I forgot the word. You have a, thank you. And it's heart rate and in sense of your heart contractility, dilatation of your skeletal muscles, and a bronchodilation. And we have these consequences: blood pressure, temperature, and alteration in the respiratory rate. And what about dental anxiety? What this is related? So dental anxiety is a specific type of anxiety that is related to the dentistry. And this affects something around 36% of the population, and 12% of the people around the world suffers with extreme fears from dentistry. And then we want to understand this. And this, we are going to think about surface temperature. And surface temperature, it's used to detect a lot of states in medical environment, like tumors, infections, inflammations, and they look like hot spots in these thermographers. So, and also facial thermography has been used as an indicator of anxiety, and then to dental anxiety. Thinking about this, we want to understand how this develops, and we are using twins to understand these mechanisms. So our aim is to analyze the reaction of twin children aged 7 to 12 years old, but in fact we have a higher range of ages, but I'm going to talk about the 7 to 12 years old children today, and check the differences between twin siblings in the reaction to this dental experience, and if there is a correlation between the dental anxiety and the changes in facial temperature. So what we use? We are studying these twins using a thermographic camera, a self-report questionnaire, that is this questionnaire that is specific about the dentistry environment, that children has to answer how they are feeling about going to the dentist, or if the dentist touches you, or if they put you that little engine, or to have to extract your tooth, and they point to us how they are feeling in these phases. And we did this in here. I'm going to show you the two situations. The first time they go to the dentist and the subsequent visit. So the first time, it's a clinical exam, just dentist looks, okay, it's okay, everything's okay, you are going just one more time. Oh, okay, you have a lot of problems, we are going to see you some 2 or 3 times more. And after that, the first treatment visit, when children are experiences for real the treatment. And so they first answer this questionnaire, and then They sit in the chair, and we took pictures. These pictures, we name it at pre-treatment, during the treatment, and after the treatment. And we are specifically looking at the tip of the nose, your forehead, and your ear. And we calculated differences between these areas' temperature before and during the treatment, and before and after the treatment, and compare it with your anxiety scale. So until now, it's until now because we have almost 70 pairs, but I bring here only 13 pairs for you. So they are around 9 years old, and the tip of the nose, it's the more expressive changes in the temperature, around 2 degrees. And we had no differences between monozygotic and dizygotic twins. And we can see here that the difference in the tip of the nose, it's the most higher. And what we can see until now is that the anxiety that children expressed in the questionnaire in the first visit was a moderated anxiety, and until now, with these 13 children, we cannot see the differences. But we can see here that it's a trend line that this could be possible. But when we look to the changes in the temperature, we see that in the second one, that is when children are experiencing the treatment, it's higher. It's a higher difference, which means that the tip of the nose is colder during the treatment than there was before the treatment. So thinking about that fight or flight situation, you are putting your blood to your legs if you need to run, and your nose is not so important in this moment. So we saw this, that there is a tendency to increase this dental anxiety in the second visit. And we can see this, expected this, because they are already showing these higher changes in temperature. Previous studies have shown this association between high levels of dental anxiety and traumatic dental events. But on the other hand, there are other factors that could affect this dental anxiety, like vulnerability, personal perceptions, the perception of that I don't have the control, what this person is doing in my mouth. And this could be related to this anxiety. And think about that. Dental anxiety, like I told you, is a response to a danger or a threat situation. That is that person that is putting their hand in my mouth. And what we want to do now is to see if age, sex, environmental sensitivity, and the repetition that we can see a learning processes to the dentist environment could change this dental anxiety. And this is a project that we are doing together, the Psychological Institute, Medicine Hospital, and Dentistry Institute here at USP. And I think this is that. These are some moments with our children, the team, the higher cameras here, and all our sponsors. And if you look at this, our website, maybe you can see something about this, all related projects. So it's that. Thank you. And if you have any questions.
Hey, thank you for the talk. Just one quick question. So what do you think the practical applications of this would be? So if you find that this is a consistent way of identifying dental anxiety, how could that then be used in clinical settings and et cetera?
I think that we could use this to reach a more humanized treatment. There are a lot of mothers sometimes tell us histories that, he felt and the dentistry was not very nice. They just put the instruments there, and my children now doesn't want to go to the dentistry. So I think that in the clinic, we can use this to reach a more humanized treatment. And this is something that in this big project we are reaching. I have a colleague, another postdoc colleague, that she's studying the dentists. So we are how they feel with different children, different responses. That children that's only cry, 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 and you just, hello, and children are already crying. So how to think and how to organize all this chaos, because it's really a chaos. It's a lot of children talking at the same time, but it's very interesting. So for the clinic, I think that's our goal, our main goal. It's just about the clinic that you asked?
Well, I'm generally practical applications, because it seems maybe the value is you're able to detect this anxiety without verbal communication, right? So they don't have to express anything. It's just you're taking these, physiological indicators of what their experience is. And I'm imagining how, because everyone wants to know how that kind of research would be applied. That's where all the money is, right? And so I'm wondering where the next step in application would be.
Yes, what we can see in this, when we look for correlations between the temperature and the inserts, sometimes younger children, they answers, oh, I'm happy. But when we took the picture, they changed 2 degrees. That is a lot of degrees, because this study was already did with adults, and that adults that has the higher anxiety levels changes 1 degree. And children, no, I'm happy. It's okay. She can took my tooth off, and she changes 2 degrees. So I think that we can understand the perception of the emotions and the development of these perceptions. Because we have children with 4 years, and we have children with 15 years. So we are accompanying them through 2 years, and we are going to be to look to the changes and all these constructions.
Hi Lilian, very nice presentation, I really liked it. I'm not sure if I missed it when you presented, but is dental anxiety correlated with general anxiety or with the other types of anxiety you presented?
I don't know how to answer this because I don't know. I really don't know. But I think that maybe, because it could be really, if it is related to environmental sensitivity, so it will be related with general anxiety. So this is our next step, to see these relations to these different type of people.
Oh, I see. It kind of sounds like a phobia, but it's might not be. Is it common in children to be? How prevalent is?
Dentistry phobia.
Yes.
They are not participating in our study, but we see a lot of children screaming, and mothers need to help to keep the children in the chairs, but they are too young to answer to our questions. So we are seeing just their behavior, and we see they are. I saw one biting the dentist, and the dentist was just there. So I think that it could be common, and this is one of the things that we want to understand. If your family has this, if it's more frequently in children too, so this is our steps that we are analyzing.
I see. That makes sense. Thank you very much.
So thank you very much for nice presentations, for a nice session. So